Not, I'll turn it over to Carol, who will introduce today's speaker.
Okay. So I'm very excited to introduce Emily Taylor from started off already, Cal Poly San Luis Obispo. And as many of you know, Emily was a undergraduate here. It's going to work, there we go.
What's the saying?
Yes, really, her and you, really, Carla. And here's Barb Stein, too, and a bunch of other undergraduates who we don't know who they are. So this is proof that MBZ Beards did happen 20 years ago. And Emily also was one of our first undergraduates, I think, that really worked in the collections as a curatorial assistant. And I have some excellent examples of her writing. This is like, tells you, so these are the catalog cards, and we still actually use these a lot. We don't write them anymore, but we go back and check data against them, and this is like, her writing is spectacular. Like, you cannot get any better than this for going back and checking data, which is, of course, something we like doing. So thank you, Emily. So in addition, an interesting fact is that she was actually an English major when she was an undergraduate here, and I don't quite know how you got involved in curatorial work, but if you can tell us about that. And she actually did an undergraduate project while she was a grad student, or an undergrad still, on Crotalus enyo, the Baja California rattlesnake. She did a whole diet study when she was here as an undergrad, so that was her first publication. And she worked some of that time with Dale Donardo, and then at the time Dale was a PhD student, but also the vet on campus, for those of you who remember him. And then he went on to become a professor at Arizona State University, and that's where Emily went and got her PhD with him and graduated in 2005. And then she's been at Cal Poly ever since then. And so Emily has published a ton, and she's really very well known for her thermal ecology and research on glucocorticosteroid in vipers and snakes and lizards, and has written several book chapters on this. So she's really one of the world's experts on this aspect of snake and lizard ecology. And she, another very amazing thing, she's won numerous Emeritus Teaching Awards, but also one of the big ones that she's won is from all the herpetology societies. She's won the Emeritus Teaching Award in 2016. And she's done a lot of service to the society, so that's actually another way that I know her, through ASIH and through SSAR, the two big herp societies. And she's gotten numerous grants through the U.S. Air Force for doing her research in Southern and Central California, and also through the NIH, through the Bridges to Baccalaureate program, so bringing in even more undergraduates to do research at Cal Poly. And a couple other sort of cute or funny factoids I wanted you guys to know. So she was part of the last class of Harry Green's that when he taught herpetology. And so 104 and in Harry Green's class, they had sort of this habit of shrieking or mooning the teachers. And she was part of the group that did that over at Kelso Dunes, that many of you who are around then would know about. And she also just opened a brewery with her partner called Seven Sisters Brewery and Brewing Company. So if you guys want to go down and see her, you can also see her there, in addition to all the research she's doing and all the awards and things that she's doing. So, and one other factoid, her sister, in addition to being an English major, her sister is also an excellent writer, and is a New York Times young adult best writer. So yeah, she's won numerous awards and is a best-selling writer. So you have a lot of talent in your family. And I did want to mention there's a talk. She's giving another talk today at 3:10 on how to get a job at a non-R1 university. So for all the postdocs and grad students and everybody out there, I definitely recommend you guys going to that at 3:10. So yeah, so without further ado, here's Emily Taylor. Thanks, Emily.
Thank you, Carol, for that awesome introduction, and thank you for the invitation. I really just can't explain how exciting it is for me to be here. I sat here 20 years ago. I was an undergrad, of course, and watched all of the awesome speakers come through every week, and I literally fantasized about being a speaker someday. So when I got the email asking me to come, I'm not a very emotional person, but I got a little bit emotional. And then yesterday, being able to walk around campus and go to a bunch of my old haunts was really cool. Cafe Milano, Cafe Strada. I hung out there all the time. And so it's been really fun to be back. So thank you for bringing me back here. I'm excited to talk to you guys about some of my research. First, really quickly, I'll point out something funny. I was talking to Carla today. There's the misspelled San Bernardino. I actually went through, I thought I was an English major, I thought I knew everything, and I literally changed the spelling of San Bernardino in everything from the actual tags of the specimens to the actual online database before I realized I was spelling it wrong and had to change them all back. This one apparently didn't get changed back, so sorry about that. My legacy. It might have been neat, but I spelled it wrong. Okay, so I'm going to be talking to you today about some of my research I've done in the past 3 years on a new system, which is mainly thermal ecology of lizards. And I'll tell you a little bit about the story about how I got involved in that, because most of my work until then, and in addition now currently, is on vipers, like Carol was mentioning. And in particular, I study the endocrinology of vipers. But this work recently is specifically going to be focused mainly on lizards, and we're starting to focus on ways that we can harness new technology to be able to get more accurate readings and what these might mean to conservation biology. Cal Poly is an engineering university, and so I'm lucky to have access to engineers who can help me with everything. So side note, right now I'm working on a really exciting project with the materials engineers to be able to artificially elevate hormone levels in free-ranging animals. Really exciting stuff that we're doing. And for this work, as you'll see, I had access to 2 full-time engineering technicians who their department is called Technical Imagineering. You imagine it, you dream it, we build it, is what their motto is, and they can build us anything, and it's really awesome that I get to do that. So that's what my talk today is going to be about. Okay, so everyone here probably knows this famous graph that you can see in most every single textbook, showing you the relationship between ambient temperature and body temperature in endotherms and ectotherms, right? So ever since taking IB 104, I can't even remember why I took the class, but it changed my life. It made me want to become a scientist. I was fascinated by these guys, by the ectotherms, right? And initially my research wasn't on the thermal ecology, but I became more and more interested in it as time went on because of these side projects that I was doing. And I'll first of all, say up front with something that most people in this room know, but my students don't know, is that we're talking about ectotherms, not cold-blooded animals here, right? And so clearly many of the lizards that I study attain body temperatures that are far higher than anyone in this room, than any endotherm. But we're talking about organisms, ectotherms, whose body temperature is dramatically impacted by the ambient temperatures around them, that obtain most of their heat from the environment around them. And my dissertation work at Arizona State, after I left Cal, was on western diamondback rattlesnakes, and I studied specifically reproductive endocrinology in them. And so here we've got a courting pair. We have a male attentively chin rubbing, if you could see it in real time, on this female who looks like she's trying to get away from him. Okay. The point is that these are really large animals, and part of the reason that I study them as a physiologist is I can get lots of them, and they're numerous, and also they're large, so I can get lots of blood from them. I can do lots of things with the blood. But another thing is that physiologists like to put stuff inside animals. As these technology has been miniaturized, we're able to actually put these radio transmitters and data loggers inside these animals and get insights that we never could before. And so while I was studying reproductive endocrinology on these rattlesnakes using radiotelemetry, I would also say, well, let's just put a bunch of other stuff in here. And specifically, I started studying these animals in the field right around the time of a major new technology. As if these miniature radio transmitters that were developed in the 80s weren't enough, in the 90s we started to be able to use these I-button temperature data loggers. Okay, this has been huge for thermal biology. To give you an idea about what size these are, they're about the size of a dime, a little bit thicker, and these were originally developed to use in cross-continental shipping. So if you're shipping produce, you would program one of these to collect body temperature every 5 minutes, every 10 minutes, every hour, whatever you want. And then it could be downloaded at the end, and you could show that your produce was never exposed to extreme temperatures. Of course, us physiologists said, well, we can put that inside the snake. And all we do is we glue a suture onto them, and then dip it in this Plastidip and sterilize that, which helps prevent the animal's body from reacting to the metal. And we can we can then suture those sutures into the animal's rib cage so that it doesn't travel up the body. This is all surgically implanted inside the animal's body. And these are big snakes, so it has very little impact on them. And during my work, I was doing all these little side projects on thermal biology. So some really cool things that I'm just going to briefly mention came out of that before I tell you the story of how I got into lizard thermal ecology. So one of the things we did was we were actually able to use body temperatures of free-ranging Gila monsters and rattlesnakes to be able to detect later on when they were active. So that was really cool. This one particularly, we were saying how important is it to go out and continually measure body temperature using an eye button versus like if you went to check once a day using the previous technology. And we were able to show in this one that checking once a day gives you dramatically skewed results. So these eye buttons are able to give us a really good idea about how the animal's body temperature changes in the middle of the night, for example, when you're not visiting the field site. Here's the one where we could use their body temperatures to detect when they were surface active, which is incredibly an incredible technique. When you can't be in the field all the time, we can actually go back, and in these ectotherms, we can say when they were active, even when we weren't there. And then finally, a more recent one is some of my students have become very interested in conservation physiology, and in particular, I talked a little bit about this in my herp group, it's about translocation, which is a common management technique for nuisance vipers. And to what extent do these animals, are they impacted by translocation to where they, for example, can no longer find suitable refugia, and we were able to show the thermal effects of translocation. So all the time while I was doing this, I was thinking about thermal biology a little bit off to the side. And then the seminal paper came out in 2010 that put it really front and center for me. And this paper was by Barry Sinervo and a huge crew of colleagues. And they took tons of data on lizard species from across the world. And they used data like their preferred body temperature and thermal tolerance data, specifically a variable called the critical thermal maximum, which is the rest of my talk is going to be centered around that. And I'll define that more formally for you in just a minute, but it basically is the upper temperature that an animal can reach and still survive. Okay? And they used those data along with the projections that we have for climate warming, and they showed that as many as 30% of lizard populations throughout the world are at extreme risk of extinction. So it depends on where you're looking and the actual thermal physiology of these animals, but in this particular group of animals, for example, you can see that this red is some populations are at risk of greater than 90% risk of extinction. So this paper scared me a little bit, and I started to think about some of the aspects inherent in this paper. And it made me think about the critical thermal maximum a little bit more detail. So here's our formal definition for the CTmax, or critical thermal maximum. When you heat those lizards, for example, when ambient temperature gets high, there becomes a certain temperature at which it becomes too hot for them. And so what this means is that they would lose their locomotor performance. And if you don't immediately cool them, they would then die because they couldn't get away from a predator or couldn't get away from further heating. Okay? And so this variable was used to create those projections that Barry Sinervo used. So this is a fundamental variable, and I started really thinking about this variable because as a physiologist I thought, how accurate were those data that we were collecting? And furthermore, what is causing this? What is actually causing these animals to stop losing their locomotor performance? This is questions that no one has ever asked before. So it all started with an undergraduate of mine who was going to La Selva for an REU in Costa Rica. And his advisor for the REU, who was a friend of mine from back in my grad school days, Stephen Whitfield, had previously shown that we were seeing an unprecedented decline in lizard abundance at La Selva. Okay? And so the primary forest is where they were seeing the decline over the years. They actually saw a little bit of an increase, although not super significant, in abandoned cacao plantations, probably because there was more access to sunlight. Well, the crazy thing about this is primary forest. These are places that are not being allegedly impacted by things like pollutants and all these other things that are impacting these animals. And so Stephen Whitfield wanted to know why might these be declining, and he postulated that climate change could be partly responsible for this. So my student went down there and did this project that really got the ball rolling for my own research. Okay? So again, this was an OTS, undergraduate research project that he did at La Selva. And at La Selva, if anyone has been there, has anyone been there? Okay. As you know, they have, I've never been, but I've gotten the nice virtual tours. As you know, they do have research labs, but the technology is very limited down there. You really have to make do with what you have. And so my student, George, who's now a PhD student at Arizona State, would collect lizards, various different species of lizards, and to measure their critical thermal maximum, he would put them into these containers, these plastic containers, right here, and heat the water. And then he would flip the lizard over, and when the lizard no longer was able to right itself, he'd measure that body temperature as the critical thermal maximum. It's worth noting that this technique is what's been used for 100 plus years to measure the thermal tolerance of lizards. And I want to point out that while this is something that's really, it was great work that we did at La Selva, it was really difficult to get the heating rates constant, really difficult to be accurate with respect to the body temperature. You had to pick the lizard up, it changes its body temperature, and so on. Okay, so these are all the lizards that he studied, having reached their critical thermal maximum. And as soon as he would take them out of these containers, he would immediately cool them, and they were able to right themselves, and we put them back. They're fine. This work was featured as a featured student, undergraduate student research in Ecology, and it made the cover, although very fascinatingly, this is not one of the species that we studied. Don't know how that works. Okay, so let me show you some of the results from this study. What we found was that if you group the animals by habitat type, we can see a difference in CTmax based on habitat type. And this is something that has been found before in other systems. There are lizards that I'll describe in more detail in a little while, things like iguanas and basilisks and amoeba lizards, kind of like our local whiptails, that like to live out in the open in the sun. They tend to have higher CTmax. And then this interesting group of lizards that lives in the buildings. These are geckos that have kind of secondarily moved into the La Selva area associated with when they built those buildings, that have a relatively high CTmax, even though, as you'll see, the buildings don't get very hot. And then finally, forest floor and leaf litter animals, things like skinks and small lizards that live in areas that are not associated with higher temperatures that tend to have a lower CTmax. All right, so those are the CTmax data that he collected, but he went a step further and he said, okay, well, let's look at what temperatures are available to these animals. And he built, again, being in La Selva, relatively crude operative temperature models. And these are going to be copper pipes, so these ones were used for small lizards like the skinks, and then large lizards like the iguanas. And this just shows you some of the ones they were doing for a separate project on frogs. There's the eye button. Each of these copper pipes has an eye button inside of it. And it's designed to model, it's a biophysical model that shows you what the temperature of a lizard could be if it wasn't thermoregulating, just sitting in that environment. And again, it's very crude because it's not shaped like a lizard, but it's the approximate body size of a lizard. He put these out in all the places that the lizards were seen active, and was able to get thermal quality of the environment. Okay, what are the possible body temperatures that the lizard could attain in those environments? And then using the critical thermal maximum data along with the possible body temperatures, we're able to see, okay, how much time can they be active now? And furthermore, with projections for climate change, how much time will they maybe lose in the future? And these were the data that he used for climate change. There's some interesting data out there that will show you differences based in dry season and wet season all across the world in Central America. And there's these kind of conservative projections saying that it's probably about half a degree all the way up to the projections of like if we don't do anything, like if we don't join the Paris Accord. You guys get my deal? Syria does, everyone but us. Anyway, the point being that there's this range of temperatures for warming, and we kind of took the low end and the high end to create two different models based on what might happen. Okay, so I'm going to show you some data now. These are our open lizards, right? These are lizards that like to live out in the open. And let me orient you to this first graph before I go through the rest of them more rapidly. This is like a given day, although our data analysis is based on many days. This is just showing you a given day, okay? And what you've got is you've got the temperature, and each one of these right here is the critical thermal maximum for the different species, okay, of those three. Right up there, these are the highest ones, right around 40 to 42. The black line shows you the current temperature as measured by operative temperature models that are out there in the associated size. And then we used an equation to estimate the temperatures, the operative temperatures and how they would change with both the low prediction model, the one that predicted about 1 degree Celsius increase, and the high prediction model, the one that predicted about 4.5 degrees Celsius. And so then we said, all right, when they cross this line, it means the lizards can't be active. So currently right now, for example, in this particular graph, there would be a short period of time on that black line where the lizard couldn't be active out in the sun because it would be above its critical thermal maximum. All right, so open lizards can lose up to 4 hours per day. What this means is that under the high prediction model, and we calculate the amount of time here, there could be up to 4 hours per day that these lizards might not be active. Now bear in mind there's a lot of assumptions here, right? There's a lot of assumptions that the heating is going to be equal throughout the environment. A lot of assumptions that these operative temperature models are accurately measuring the biophysical properties of the lizards. This is to give us a general idea and to compare across habitat types. Because look what happens when we start to look at different species of lizards. This is one of the most interesting parts of the study, I think, where we found that these geckos, the ones that live in the buildings, appear like they will not be impacted at all. And that's because their critical thermal maximum is so much higher than that of the environment where they live. Okay? So they're all up here, and the buildings are really low temperatures, okay, where they hang out because they're out at night in these temperatures, right? So even throughout the day, they're going to be in shaded areas. And even the high prediction by adding those, these animals are not going to be approaching their critical thermal maximum. All right, let's look at some of these low critical thermal maximum animals that live in the forest, like our skink, who's our leaf-litter lizard. This one could lose up to 9 hours per day, only under that high temperature prediction, right? So the low prediction wouldn't be much different from current, but the high prediction one means it could lose up to 9 hours per day. And again, I'm showing you one representative graph, but we used a bunch of different figures and a bunch of different days to get these data. And then, of course, the most dramatic one is our forest floor lizards, where some of our forest floor lizards, these two species down here, under the high prediction, the forest floor could actually warm to where the entire day could be above their critical thermal maximum. Again, the assumption here, this is above ground. Some of these animals do dig down into the forest, into the floor, underneath the leaf litter. But the point is that this could be bad for some of these lizards. So in fact, the changing climate already could be responsible for some of the declines that we're seeing in La Selva lizards. So George did this work, and then I helped him analyze the data, and it really got me interested in thermal physiology, because I started to think, why are these values so different from species to species, this critical thermal maximum? I mean, clearly it represents natural selection based on their habitat type, but why? And as a physiologist, how? And so I started to answer those questions using the western fence lizard, which is locally, probably here too, down in San Luis Obispo. As a physiologist, you always say, what's the most common animal? So I study rattlesnakes and lizards because they're the common ones. And my students and I can just walk outside the building and catch a bunch and bring them inside to work with. And our question that we wanted to ask was, what is the actual physiological mechanism responsible for the CTmax? It's been bantered around a little bit in the literature. Things like the muscle, myosin ATPase stops functioning at a certain temperature, or perhaps it's the nervous system. It's not the muscles themselves. Maybe it's just the actual coordination of the central nervous system. And one of the hypotheses that I found out there that was mainly in the invertebrate literature I wanted to test was this oxygen and capacity limited thermal tolerance. So let me explain this hypothesis to you. This is the idea that as the body temperature of an ectotherm goes up, so does this metabolism. So it's using more and more and more oxygen. And then at some point, its demands for oxygen in its muscles and its nervous system, for example, to right itself in response to being turned over, exceed the ability of the circulatory system to deliver oxygen. They just cannot get enough oxygen. So are they going like hypoxic internally, basically, at the time period at which they lose their righting response? And so I'm going to go through a series of tests that we've done. It's worth noting that all of this was done by undergraduates in my laboratory who were able to do what I consider to be master's level research because I got them when they were freshmen. Recruited them out of my intro bio class. And both of them just graduated and are getting ready for grad school now. Okay, so that's Mason on the left and Tanner on the right. And this was our first device. Okay, so this device here that I'm going to show you in more detail in just a minute was a Cal Poly engineered device. And we're really excited by it because it allowed us to get rid of all the problems that were associated with measuring critical thermal maximum in the past, so we thought. Things like the thermocouples that are used to measure the body temperature have a plus or minus 2 degrees Celsius accuracy, which is really not good when you're trying to look at minute differences. The heating rate can dramatically affect the actual critical thermal maximum, so we wanted to make sure we had standardized heating rates, not like the La Selva style where you had to depend on water baths. And furthermore, we wanted to be able to test the lizards in the field right after we caught them to control for any acclimation effects. So this is what this looks like. We call it CROX, the Controlled Reptile Oxygen and Climate System. Because the last thing that we wanted to be able to do with this, yes, we definitely matched the name of it to the acronym. Another thing we wanted to be able to do with it was to be able to alter the oxygen composition. Because we're going to be testing this oxygen and capacity limited thermal tolerance, we wanted to actually manipulate oxygen so that we could look at the effects on the critical thermal maximum. So let me show you a diagram from the paper to see a little bit better what was going on with this. Basically, this tank is an oxygen tank that is full with a different gas composition. It could be ambient air, or it could be hypoxic gas or hyperoxic gas. And this whole system is set a series of valves, and it will go through a coil that's surrounding a heater to where the air is going to be heated. Okay? And it goes through a system. This is going to be connected to a laptop over here. And there's an Arduino, basically an Arduino situation set up to where it can turn off or turn on the heat flow, so that we can maintain a constant flow of heated air into all of these chambers. And then each lizard is fitted with this is actually an RTD, a resistance temperature detector, that has a plus or minus 0.5 degrees Celsius accuracy. I love our engineering people who figure that out. And then those also go back to the system to be logged on the laptop. So essentially we're able to heat our lizards at a constant 1 degree Celsius or any other temperature that we want rate, which is kind of a standard in the literature. And these are tubes, so as we're heating them, we can turn them over constantly to see if they right themselves. So this is a diagram of, or a video of us in the field, and the students, when the lizard starts to get hot, are like just flipping them over. And then the lizard will eventually not be able to right itself, and we can just click and log that as their critical thermal maximum. And this is a hint for future studies, because you can see we're doing this in the field. It's really cold out. We're up at the top of the Sierra Nevada, about 9,000 feet doing this. And I'll tell you why about that in just a minute. All right, so experimental design for this first study was we exposed, this was not at the top of the Sierra Nevada, this was at campus. We would go out and collect the lizards and expose them to either normoxic gas or to hypoxic gas. And we chose this based on similar studies that have been done looking at different responses in lizards. And then hyperoxic gas. We chose that based on the highest amount of oxygen that our safety people at Cal Poly would allow us to do, fire. And then we would heat the lizards while they're being exposed to these different types of gas, and of course wait until they couldn't turn themselves over anymore, and that was the CTmax. This is my very high-tech diagram of our experimental design. Okay, so this is one figure that kind of encapsulates a bunch of our results. Let me take a moment to go through it. There's a body size effect, which is why we're showing our covariate of snout vent length on the x-axis, and then here's our critical thermal maximum. The data that show the normoxic are shown in white squares going across here with our dashed line. No significant effect of snout vent length in that particular treatment. And then hyperoxic is with black above it, and hypoxic is below. So the first thing I want you to notice is that we see a very strong treatment effect where the post hoc tests showed that the lizards exposed to a hypoxic gas had dramatically lower critical thermal maximum than the lizards exposed to either the normoxic or hyperoxic gas. The second thing I want you to notice is that snout vent length is a significant covariate only due to the hypoxic treatment group. So the larger the lizards got, the lower their critical thermal maximum was, only under hypoxic conditions. Okay? So we've done follow-up work since then that I'm not presenting today on that body size effect, because one of the reviewers was freaking out about the body size effect, saying it was totally unprecedented. Because we were thinking that in support of our hypothesis, it could be that, you know, if the oxygen has to travel further through the body, despite the fact that their mass-specific metabolic rate should be slightly lower in a larger lizard, having to deliver that oxygen longer distances could lead to a reduction in the critical thermal maximum. But in reality, I think it might be an artifact of a difference in heating rate. So since then, we've gone out and we've collected 4 different species of Scoloporus that are at much dramatically varying body temperatures, and showed that even under normoxic conditions, the big ones heat more slowly, like you would expect, and therefore have a lower critical thermal maximum. I can talk to you guys about that later on if you'd like. But the important thing for this is that we showed this hypoxic effect. And with regard to hyperoxia, we didn't see, the post hoc test didn't show that the average differences between normoxic and hyperoxic were different. But if you actually take the typical critical thermal maximum, the highest value you would get under normoxia, under normal conditions, which is about 44, you can see that there's actually a much higher number of lizards that made it beyond 44 when they were exposed to hyperoxia. So we did a chi-square with that and found that that was significant. So this is to be expected. You know, you're not going to, when you're exposing lizards to the critical thermal maximum, their upper lethal temperature is just above that. So you shouldn't expect, even with hyperoxia under this oxygen-limited hypothesis, that it would go much higher than that. But we found some support there. Okay, so the next one that we did was taking it to the field, where we chose to actually manipulate oxygen at different elevations because of the effect that atmospheric pressure has on partial pressure of oxygen. Okay, so for this we used a transect that we developed in the Kern River Gorge at the southern Sierra Nevada. And for this study, we took it to the field. So this is the exciting thing, because at this time, and still to this day, everyone would go out to do similar studies, including most of the studies covered in Barry Cinervo's science paper, would go out and collect animals and then keep them in the lab for a while before they did this. So we were actually able to do them on the day that they were caught, but also at the actual ambient temperatures and ambient oxygen conditions. So we basically just have the system all attached to a generator over off screen here, Those are socks right there, which the lizard bags are socks. So they're right here. So each one of those is a numbered sock. And there you can see the generator and the compressor that we had to take with us into the field. This will be important later when I tell you about the next generation crocs that doesn't have to use this. And we would collect our data. And our study species here over the transect we actually went there expecting to just study Occidentalis, but we found they dropped out at the higher elevations and gave rise to the western sagebrush lizard, Operative graciosus. So we studied both of them. Here's a diagram of our transect. So we had 4 sites. One was down at the river at a campground, and then that had Occidentalis only, and then we had a slightly higher with Occidentalis. We had one place at about 2,100 meters where they overlapped, and then at the very top of the mountain at about 2,600 meters we had only graciosus. Okay. So at each one of these, we would expose lizards to either the normoxic treatment, which was 21% oxygen, or the hypoxic treatment, which was 6% oxygen. We chose to focus just on those two and not hyperoxic, mainly because we couldn't get permission to bring hyperoxic gas tanks into the national forest. But also, we had already seen at this point that the hypoxic versus normoxic was the most interesting one. Now, bear in mind that as you go up in elevation, that these are going to actually lead to different concentrations of oxygen that are available. This is the point of this experiment. So we actually calculated the partial pressure of oxygen at these different sites too. So under normoxic conditions, the partial pressure of oxygen goes from about 144 down to about 117 at the height. And then similarly, much, much, much lower. Like with hypoxic treatment, not actually environmentally relevant gas concentrations. This is really low oxygen because we were trying to look at proof of concept. Does oxygen limit the critical thermal maximum? Okay, here's what we found. So this is looking at the oxygen effect in the CTmax. So we've got 2 species up there. Graciosus is shown in triangles and Occidentalis is shown in circles, right? And there's the partial pressure of oxygen. So we've got our higher elevation field sites right here and our lower elevation field sites right here. And I want you to notice immediately that at our low, step back, that's not true. These are going to be the hypoxic conditions. The hypoxic conditions from about 30 to 45. Okay. So under normoxic conditions, no matter what the field site was, all 4 field sites, we saw no effect of oxygen partial pressure on the CTmax. Okay. We only saw it when you started to use hypoxic conditions, and we saw a very strong relationship. As the partial pressure of oxygen went down under hypoxia, so as you went up the mountain, the critical thermal maximum of the lizards also went down. And so this is providing some support for the oxygen limitation hypothesis, just like our last experiment did, because you've got this commensurate decline in the critical thermal maximum the less oxygen that you get. We also saw sex difference in CTmax here. So the sex differences that we've seen throughout all of our studies are consistently showing that females have a higher critical thermal maximum than males. And this is something that hasn't been studied in a lot of detail. So this was only in Scolopius graciosus though that we noticed it, where especially at these low oxygen partial pressures, the females can have a higher critical thermal maximum. Reasons for this, possibly, most of the females were gravid, right? So these animals are gravid. They're out basking like crazy, and so potentially the recent thermal history of being exposed to higher temperatures may actually be causing them to have a higher critical thermal maximum. One more thing on this study before I tell you about the last series of studies that I'm going to talk about today was we wanted to look at species differences in this one area of sympatry where they're both going to be on the hill right there. So we looked in there and we found that there was a species difference, that Scolopius graciosus had a much higher critical thermal maximum than Occidentalis. So graciosus are shown here, and this was especially in hypoxic conditions. Okay? And so this may be partial adaptation for living at high elevations where these animals are better able to deal with hypoxic treatments. It may also be a body size effect, like we saw before, because graciosus are so much smaller than Occidentalis. Okay, so the final study, which is actually brand new data that my graduate student is doing now, was we started to see variation in critical thermal maximum. And our reviewers were saying interesting things like, for those 2 papers, they were saying things like, well, I don't know why you're looking at how sex and body size impacts the critical thermal maximum, because it's just a species-specific value. And we thought, but it's not. Saw that it wasn't. And so we started to think, okay, is it affected by acclimation? Do these differences that we see go away when you acclimate the animals? Or better put, is that paper by Barry Cinervo, are all of those data viable, given that they're mostly done on animals that are kept in captivity for a long period of time? So If our answer is no, that they're not viable, then we're probably in big trouble and we're unpopular biologists because we're saying that acclimation can affect body temperature. On the other hand, if we don't see much of an effect of acclimation, then we can say, oh, these data are good, you can keep animals in captivity and it's fine. Okay, so actually I'm running out of time, so I'm going to skip over this. This was the study of interest here. How does acclimation to lab conditions affect the CTmax? Okay, so this is our next generation. I always just turn the students loose to try to work on the acronyms themselves. So now we have the gas analysis, temperature, and oxygen regulation system, which is nice because all it needs is a power source. We don't need to have air tanks. It can heat the lizards and it can cool them. And we have minimized some other problems that we had, like surface contact of the lizards with the actual hot tubes. And so these data are, and also this is a tube within a tube. So it just spins. So you can just turn it and it spins them, which is really nice. This is also field portable. Okay, so this is some brand new data that my graduate student just gave me last week. And what he did was he set out to look at the effect of acclimation and season on CTmax. And he tried to do a couple different acclimation trials to mimic what might happen when scientists go out to collect animals. So one condition we thought of is they collect animals and they just leave them in bags in the lab for 2 days. Because we do that all the time. Right, when we're doing stuff. They just in bags for 2 days and then we test them. And then the next one is we put them in a cage for a week. So we'll get to that in just a minute. So he did it in spring, summer, fall, and winter, and we had some interesting but very paradoxical results. So first of all, non-acclimated lizards were done the same day in the field. So those should represent their actual CTmax. And then a 2-day acclimation. And what we can see is they're very consistent, the 2-day acclimation, except for in the fall. I don't know why that is entirely. In addition, we can see that if you just look overall at the seasonal effect, we did see a seasonal effect where the summer animals had, contrary to what you might think, a lower critical thermal maximum. So, okay, so maybe we're seeing a lab acclimation effect, or maybe we're seeing a seasonal effect. This was confusing data, so we're repeating it. We're just doing it all over again for the second year of his study. Because he also did some, yeah, the winter. It was really hard to find lizards in the winter. So we didn't have enough to do the acclimation in the winter. So we're hoping to get out there and dig them out from under rocks and do a better job this coming winter. The other thing that he did was he took them into the lab and put them in environmental chambers and acclimated them for a week at either kind of room temperature, hot, or cold to see how does that affect it. And okay, got interesting results here. Here's our same data from before, the summer same day and the summer 2-day acclimations, which were relatively low. And then look, no difference between our 15 degrees C, 25 and 35 degrees C. Much higher critical thermal mass. So if you just look at this by itself, because these were all done in the summer, right? These were done on animals that were collected the same days as these guys. If you just look at this, you would say, oh, well, a week in captivity, no matter what the temperature is, increases the critical thermal maximum. But 2 days doesn't, but a week does. But then if you go back to our previous slide, we saw that actually their critical thermal maximums tend to be up around that, and it was just depressed in the field in the summer. So what's going on with that? So that's what we're working on now, is trying to wrap our brains around that, and again, trying to replicate it, because these are the types of studies that can be replicated and should be replicated. Okay, so our next study is repeating acclimation studies. One of the things we're working on now that I'm really excited about is we're using a next generation lizard OTM, so not the La Selva kind. I'll show you what we're doing. To actually predict how a common species like Occidentalis may be affected by climate change. So here's a little diagram of some of the really cool OTMs we can make nowadays. My student took a dead lizard and made a lizard wax model, and then took that wax model and using some sort of magic, that will escape me because I was an English major and I only took one chemistry class, he was able to actually coat that wax model. So the wax model has an eye button in the center of it. Coat the wax model in copper solution, and then it hardens into copper, and then you can just merely melt the wax out of it. So now you've got a perfectly shaped copper lizard with an eye button on the inside of it. So cool. And so this is us calibrating it in an environmental gradient. So that's an actual lizard with an RTD in its cloaca, and then there's a copper lizard with an RTD in its cloaca, so we can look at how well it actually estimates the temperature. And then we deploy these in the field. And so the ones that didn't get picked off by hawks, which happened a lot, because they're very realistic, they'd sit out there for the whole year. We put them on trees, we put them in rock crevices all around so that we can get an idea about the thermal landscape. So those data are still being downloaded by my students. I don't have those to present today. In addition, the new gators can do critical thermal minimum. So we are all of a sudden super popular. Everyone wants to work with us because we can go out into the field, measure critical thermal maximum and minimum of any species. For example, this summer we're going to start working with asexual whiptails on an elevational gradient in the Chiricahua Mountains, looking to see whether those unisexual whiptails actually have a trade-off associated with asexual reproduction that's being found in a lot of other animals. Maybe a thermal trade-off. And then finally, we're starting to refine our data to be able to actually measure blood oxygen saturation and metabolic rate, both of which now are possible at Cal Poly. Our new gators, all you have to do is hook it up to a multi-channel stable system, and we can get their metabolic rate as they're being measured, which is pretty slick too. My current grad student is exploring how disease, specifically malaria, which is a common disease of fence lizards, actually impacts their critical thermal maximum, because that disease makes them anemic. And if anemia lowers their oxygen carrying capacity, then potentially we can see an effect there. Okay, so I'm glad I got finished in time for some questions. These are some of my many, many students. I have a big lab at Cal Poly. Those of you students who come to, or graduate students or postdocs who come to my talk later on about how to get a job at a non-Research One university, I can tell you a little bit about how things work at my school, which is a master's granting institution. It's pretty great. I get to work with a ton of awesome undergrads and grad students. And our funding came from a number of different sources, and here's our guy from Technical Imagineering who helped out so much. So, thank you.
Lizards acclimatize to projected climate change?
You mean like how well that they may, if you put them in long term in environmental chambers to mimic climate change?
Yeah, see if their CTmax will adjust.
Yeah, I think that would be a great project for a future student. There's a number of labs that are doing that work right now. And in addition, they're trying to model like some more environmentally relevant processes. Most of that's actually happening with things like Drosophila, though, because most of what I've done is I've said, boom, if the temperature changed like that, how would they do? But that doesn't account for actual adaptation, let alone acclimatization, over many years that will take. So that work is being done mainly in like Drosophila. But certainly in our lab, we could do something where we did a much longer term acclimation. I probably wouldn't do it with Scoloporus because they're tricky. If they're not allowed to bask, they don't do well for more than about a week. So I might do something like an alligator lizard, which is more of a I call them skulkers. Is there a technical term? Thermoconformer. Yeah. They skulk around under the bushes. So the heliothermic lizards, that is a technical term. The ones that are sun-loving, like these guys, they just don't do well in long term without being able to thermoregulate. But yeah, that's a great suggestion to do a long-term exposure. Yes, thank you.
I was surprised by the ambient temperatures in the forest in La Selva. It was like around 26. It wasn't very hot.
Yeah.
What's the elevation there?
I don't remember. I haven't been there, so I don't remember. It's decent. It's isn't it middle elevation? I don't remember.
And
I don't remember off the top of my head.
What I was wondering is whether any of those species also occur at lower elevations where it must be hotter.
Indeed, many of those species do, yes. In fact, most of them do. I'm not sure about some of the forest floor and leaf litter ones. I'd have to look that up. But a lot of those are really common animals, things like iguana, things like the basilisks, very, very common. And there are some people now, so if you remember back to my elevation work, under normoxic conditions, there was no difference across elevation, so there didn't seem to be an effect. But there's a number of people who are showing in, for example, frogs in very, very large elevational gradients down in the Andes, for example, that the ones at the higher elevations that are exposed to lower temperatures have a lower critical thermal maximum. And if you actually move them down to lower temperatures and let them acclimatize in an outdoor arena down there for a while, then it will go up. So the critical thermal maximum doesn't seem to be a species-specific value like our reviewers were telling us. It does seem to be labile and affected by acclimatization over time, at least in some species. Yes?
I was wondering if you thought of comparing your cool, fancy setup to the way people have done it before, just to get some idea about how far off the older measurements would be.
That's a great idea.
What that would mean for basically predictions from that data versus your data.
Okay, thank you for asking that. That's a great question, because I hadn't thought of it before, but we're totally going to do it. Here's why. That's exactly the kind of stuff I like to do. As an experimental biologist, I like to say, okay, what's the impact of our work? And so that's what we did. That's what initially got me interested in thermal biology, was we said with our rattlesnakes, okay, people are going out and they're collecting one temperature a day, and it's time of day when we visit. how does that impact what inferences you would make? And I did that exact comparison and found that it was ridiculously off. And so here we could actually do that by doing the same lizards or same population of lizards in the heated cup method. Okay, well, I'm going to do it. Thank you for your suggestion. What's your name?
Ammon.
What's that?
Ammon.
Ammon? All right, well, thank you. That's a great suggestion. It never occurred to me. Yeah, Carol.
Sort of a related question actually is, can you look at older values? I don't know when people started doing CTmax, but like they were published maybe in the 50s or before then, and see if Scalopers, at least Scalopers occidentalis, has changed their CTmax over time.
Yeah, that's a good question. So there's a lot of studies with occidentalis. There was one from the 70s, and the values are about the same. But it's just so difficult given the differences in methodology. It's tempting, but it's difficult. I think that what you could do it if you were doing it with a giant data set of many species of lizards, and you had a lot of species doing the same thing, then potentially you could make that. But given that we've noticed things like whether they're fed or fasted can affect it. There's a lot of things I didn't talk about today. A lot of other things that you have to control for can impact the critical thermal maximum. And so it would be hard to, I think, do that without a giant data set. It sounds like a really good maybe PhD program where someone could be measuring a lot of different species populations. Yeah.
So I guess I was thinking about the high elevation lizards and how in a lot of animals you get like hemoglobin differences that change the efficiency for carrying oxygen in the blood. Do you think that would affect, I guess, the CTmax, or and if I guess is there any? I don't even know if there is evidence of that in these lizards.
That's a great question.
Yeah.
So when we did our elevation study, as well as all the other studies, we brought a centrifuge to measure hematocrit, and we brought a hemoglobinometer to measure hemoglobin. And we never found any differences among elevations, but we consistently found the obvious fact that males had a higher hematocrit and hemoglobin concentration than females did. And so despite the fact that female had a higher critical thermal maximum than males. So that's definitely against the oxygen limitation hypothesis. By the way, I failed to step back and kind of globally tell you guys what I think about the oxygen limitation hypothesis now given this. I think that it may be exerting only a tiny influence over the lizards in general. I think it's much more important for aquatic organisms where oxygen is limited. I think that our effects that we showed, we're merely showing you the effect that oxygen had on a lizard's activity, which may or may not be related to oxygen limitation. In many lizards, many organisms go catatonic when they sense low oxygen. And so, furthermore, our lack of effect of hemoglobin. And the little study that I skipped over, we actually we made the lizards anemic and showed that it only had a mild impact on their critical thermal maximum. So I really think that ultimately, here's my idea about what's happening. I really think that it's a fundamental temperature dependence of the nervous system, which may be affected by acclimation, for example, due to changes in the membrane composition of cells and those sorts of things. But I really think that it's the nervous system. Some supporting studies have shown, and I'm not about to do this with my students, but some studies have shown that if you cut the spinal cord of the animal, that it's still, because in frogs specifically, you can actually look at the spasms that they have as opposed to the loss of writing response. That if you cut their spinal cord, they won't exhibit those spasms anymore. So it's really showing you that the spasms are originating from the nervous system, not from the muscles themselves. So that's all kind of how we're getting at the idea that this is probably an inherent temperature dependence of the nervous system that can be affected by acclimation.
Okay. Let's thank Emily one more time.